Good morning, everyone. It's a pleasure to be here. Thank you for the Alex TLC team for inviting us to be here today. So I'm going to talk to you about research, a little bit about my research, but mainly about what we're trying to achieve in the UK on the research front. So we call this network Leukolabs, and I'm going to tell you why. Karen, I think this is my old slides. I will try to make it work. It's okay. I know them by heart anyway, but I would just need to shuffle. No. So, this is the ideal scenario. Someone is diagnosed with a genetic disease. You find a mutation in a gene, and what you want to do is for this to be streamlined, and you find a therapy, and you got a healthy patient. But as you all know, this is not the case. It's really difficult. It's very long. It can take decades. So it's a whirlwind of events that needs to happen between those two, and this is when research happens. So you need to find biomarkers. How can you detect this disease? And you need to understand how the disease develops. And this is when my lab and many of my colleagues come in, trying to understand what starts the disease, how it gets to the clinical signs that you can see in the patients. And for this, we use models. So we have a lot of models in here. As you can see, we got mouse, we got zebrafish, so loads of zebrafish today, so I'm going to emphasize on that one. We got flies, but we also got cell culture, and brain in a dish. And my colleague James, who's going to talk to you tomorrow, will hopefully tell you a little bit about that as well. So then you need to develop a therapy, and then you need to test it. So all of this takes, like I said, a lot of time, and reaching this stage is having an impact. And this is where, as a scientist and as clinicians, we want to have an impact. So how can we measure impact? So we, as clinicians and scientists, we're often in that box here, the discovery box. Sorry, I move a lot. The discovery box. But then this is not enough, because we're going to discover a new disease, we're going to discover something in the lab about that disease, but all of us separate, not working together, it doesn't really help. So once we have that discovery, we need to reach, we need to engage, we need to collaborate, we need to talk to each other. And then by talking to each other, maybe we can start applying that discovery. So we can test it, we can trial it, we can optimize it, and then maybe we have to go back to the discovery box and then start all of this again. But once we're confident with whatever we discovered, what we've applied, maybe a product we developed, then we can amplify it. And then we have a sustainable impact, hopefully across many patients, across many diseases. So we realized that this didn't really happen in the UK for leukodystrophies. So we created Leukolabs. So I say we because it was many of us. Like I say, James is in the room. He's based in Leeds, and he's got Lydia Green and John Livingston. I can see Lydia. Here she is. And then there's a few of us in York. So there is myself, and there is Sean Sweeney. And you can see that by the picture of James, myself, and Sean. We've got a little model attached to ourselves. So James has the brain in a dish, I have the fish, and Sean has the flies. So today, sorry, I'm a bit biased because I work with zebrafish, so I'm going to show you a lot about zebrafish action. But the first goal of our network was to try to bridge a gap, which is when a patient is being given a diagnosis with a mutation in a gene that no one knows, no one's being studied. We want to use our quick, efficient models to develop and generate data, results, functional data, to try to understand, well, maybe give them a little something maybe this affects this pathway or this pathway. Maybe we can try this drug, this drug. The idea is that if no one is working on these genes, we're never going to know what's happening in a disease, and we're never going to have a therapy. So that was our first assay. And that's why I'm not showing mice here, because mice takes a very long time. With flies, with cells, with zebrafish, things can go very quickly. And I'll try to show you a little story, our first Leukolabs story, to illustrate this. And the second aim of the network is, like I said, to bring people together. So you can find this on our website. This is literally a copy-paste. The main aim is just to connect. Clinicians, scientists, working together, going back and forth. So building a national and hopefully also international community, and then boosting leukodystrophy research, and then promoting this translational leukodystrophy research with a patient-focused outcome. And how are we going to do this? We're going to do it together. You've already heard about the registry. You've already heard about the services. You know inside out Alex TLC. And hopefully I've introduced us, and then the four of us hopefully can try to make this happen. So like I said, I'm going to talk to you a little bit about science today. So apologies, my slides might be a little bit mixed up. So I might have to shuffle back and forth, but I'll try to make it as smoothly as possible. So this story starts with patients. So John Livingston and Lydia identified a pair of siblings in Leeds, which looked like they might have a leukodystrophy. So then he went through the genetic services to get their genome sequenced, and this is where James comes in. And then they identified mutations in the SUB-V3L1 gene. And then you would say, the what? What gene? And actually, that's what happened. In this case, there was pretty much nothing known about this gene. So we'll just call it a gene for now, because whatever name it has, it doesn't really matter. I have the list of the clinical signs, and one of the signs that piqued everyone's interest is the vitiligo. So for those who don't know what this is, and I didn't know what that was either, is when you start losing pigment on your skin, and this is associated with disease of the immune system. So when there's something wrong with the immunity. So we went ahead in our lab and tried to make a zebrafish model for this disease, because this is what we do. So at this point here, I should have a drawing of a zebrafish. But if you want to know everything inside out about the fish, we are next door. We have adult zebrafish swimming, happy. We've got baby zebrafish. We've got wooden zebrafish to draw on. We have drawings of zebrafish. We have everything zebrafish. So if you want to know more about the model, my slides aren't here, but we are next door, and there is Holly and Lizzie ready to talk to you zebrafish all day. So please don't be shy, go and chat with them. So what does that fish do? So if you target the same gene in that animal, this fish starts dying. And that was interesting. So the fish can be sick as well. But what is really going on? So this gene helps to chop down RNA in your cell. So James and Lydia said we can use some human cells, and then we can see whether we see accumulation of RNA if this gene is gone, this protein is gone. And this is what you can see at the bottom here. In the mutant, so in a mutant cell, here, there's a lot more green, so there's a lot more RNA accumulating. And apparently this gene is supposed to work in the mitochondria. So the mitochondria is the powerhouse of your cell, is what gives the cell and your body the energy to function. So we said, let's look at the mitochondria of the fish. So you can see those images here again. Beautiful imaging you can do with the zebrafish. Beautiful, they're transparent, you'll see them. So what we saw is that mitochondria are usually tubes, as you can see on the picture on the top. But in our sick fish, they are all long and mixed up together, very thin, or they start fractioning, so the cell is dying. And what we do in the lab is to look at immune cells, the immune cells of our brain, and they're called microglia. So we have a lot of different assays that we can do this. And the cells that I'm showing you here with the mitochondria inside, they are those immune cells of the brain. Again, I'm missing a slide here, but I'm going to show you this now. What we wanted to find out was what is activated in those fish. So the immune system is not working very well. There is something that's not going quite right. There's a lot of RNA hanging around. And what looks like RNA? Viruses look like RNA. So the cell, the body, is being tricked. It's being attacked by a virus, and then it activates this immune response. And another way to analyze this is to look at all of these genes expressed during an antiviral immune response when the body is being attacked by a virus. And when we saw these results, for you it's just going to look like bars, but I've done a few leukodystrophy models, and I have never seen a response that drastic. We're talking about 2,000-fold increase. It's a lot. So we decided to talk to Lydia and James and everyone in the network and show this data. We were sure it was really happening in the fish. And then we managed to get some patients that came back into the clinics to check whether they also had this reaction to the mutation. And these are not the same patient, but it's another family that was identified in Birmingham. And you can see here, all those green bars, it shows that this patient also has this activation in the antiviral immune response. So this means that there is something that we could find in the fish that we didn't have a clue about what could be happening in a patient. And we managed to validate it, to confirm it into the patient. And this means that now we could perform tests in those patients being guided by the results in the fish. And this is quite a sensitive thing to do, as you all know. What we could also do is to expand. I think half of my slide is missing here as well. But we could also do is to expand and reach out to people. And now I think it's between 14 and 16 families in the UK and in Europe that Lydia has been collecting data from. So from a pair of siblings in Leeds, communicating and talking to people, we now have 16 families that are affected, that the same gene is affected. So apologies, I'm going to try to find the slide again. I'm going to go backwards. So the idea now is that you have this gene, you have this pathway, you know what's happening, but can you create therapies? And I'm going to talk to you about zebrafish again. And the idea is that those immune cells in the brain, they are super activated. There is something wrong with them. So what can we do to target them? And this is when I was really lucky that Holly decided to join my lab to do her PhD. Again, she's next door. Do talk to her, please. And she wanted to replace those microglia. So a lot of you in the room are going to be very familiar with bone marrow transplant, which is one of the only way to replace the immune cells in your body. But Holly wanted to take a shortcut. She said the other immune cells, they don't do anything in that disease. We just want to replace the microglia. So she took what we call the progenitor cells, the macrophages. These are the cells that crawl into your brain when your brain is developing and transform into microglia. And she said, we're just going to take those cells, we're going to put them in a fish, we're just going to see what happens. So she did just that. She took a healthy fish, the equivalent of the bone marrow, with green macrophages, because in a fish, you can label cells any color you want. So she took those cells, purified them, make sure that we only had macrophages in there, put them in a tiny little needle, and injected this into the blood circulation of a baby fish that is developing. And once she saw, and you can see this is the head, the top of the head of the fish, the cells love going to the brain. Why? So during development, your brain, every brain, make too many neurons. All those brain cells, we are making about double as many as we actually need. Why? Some of them might connect, make a circuit, what we call, together. And these ones we will keep. The ones that fail at connecting, we will kill them. It means that your brain can have up to 40% of cell death happening during development. At this point in human, the baby is still in utero, inside its mum. So the brain is developing, and this is what we call a wave of apoptosis. And this is what's really important here, is when this happens, it calls in the macrophages in the body that are being made. And those macrophages, they can smell it. They can sense it, that there is something in the brain that's happening. And they are amazing housekeepers. They love cleaning. That's their job number one. So they are being attracted to the brain. They go into the brain. They become the microglia. And their first job is to go around the brain and hoover up all of those dying cells. This happens in everyone's brain. It happens in the mouse. It happens in the fish. So we decided to think, we say, right, so these cells, we've just transplanted them. They're from another fish. Do they act like microglia? So what we wanted to check is, do they actually go in the brain and become microglia? And the answer to this is yes. So Holly used markers specifically to microglia, and she could tell, yes, they look like them, and they express the exact same markers. But the question is, can they function like them? Can they hoover up those dead cells? And the answer to this is yes. I hope this is the right slide. Just going to check, yes. So the first box here, the first black box, do you mind if I move again? This one is a healthy brain. Very little number of dying cells. This one is the leukodystrophy brain. A lot of them. Because like I showed you, those immune cells, they're not functioning right. But then the green bars are when Holly transplants healthy macrophages. And suddenly that number goes down, and it goes down back to the normal, healthy zebrafish brain. So Holly was confident that by transplanting, she could rescue this debris accumulation in the brain. But then our leukodystrophy zebrafish model, they don't swim as well. They don't move as much as the control, the healthy fish. So she wanted to ask, this transplantation, can it rescue the movement of the fish? And the answer to this is also yes. I think that's not quite the right slide either. But the last two bars are supposed to be green, and the fish start moving as well as the healthy control. So this is very preliminary data. This is looking at in the baby fish, which you will see next door. Then we want to see when those fish grow, what happens to them. Do they swim better? And how is their white matter lesion in the brain? So for this, we've got different assays. So Holly can clear up the brain from all the fat and then have this amazing clear image of a brain, and she can make them spin any direction you want. So we're going to be looking at myelin, we're going to be looking at immune cells, everything that matters, oligodendrocyte that make the myelin, and be able to see whether this transplant can rescue all the cell types in the brain. And you all know what an MRI machine is. Going in there to have an MRI scan is not a very jolly experience. Well, it isn't for the fish either, but we've managed to do it. So now the zebrafish can be put to sleep with a little tube in their mouth, so they have water, so they don't dry out, and they can go into the MRI scanner for about 2 hours, get a very nice MRI scan of their brain, and then they come out and they continue swimming again. And then 4 months, 6 months later down the line, we'll do a checkup and we'll put them back into the MRI scanner again. So this is what we're going to do on those mutants. So as you can see, this was all mixed up. I changed my mind last minute about the order of the talk. So this is me finishing. So I want to thank everyone who's giving us money to be able to do this research, and everyone in the lab, everyone who works on the SUB-V3 story that I presented to you earlier. And then if you want any more information about Leukolabs itself or the research, You can come next door to find us at the break, and at lunchtime we got posters, 3 of us that can talk to you. And then thank you very much.